Okay, here's the poop. Here's the deal. I'm going to make this little video for AP Bio to accomplish a few things. I want to show you how you can do a dihybrid cross using a Punnett square in genetics. And then my goal here is to, once you've seen how to kind of set this thing up from scratch, that you'll be able to engage in an extra credit opportunity if your heart is so inclined. And so today is a dihybrid cross, but what if you had 4 traits to investigate? 4 traits with 2 parents that are both heterozygous. What would your Punnett square look like? So if you want to take on a quadrohybrid cross, you could do that, and if you watch this video, I think it'll start you down the road of the steps, even though it's more grandiose than the smaller steps with a dihybrid cross. Anyway, a couple of announcements were on the topic. We're looking at week 8 when you should be watching this video. And in week 8, the topic is genetics. And so for homework, I've got a file there with genetic worksheets, and it is ginormous. Sorry. So here's the thinking. I would like you to scroll down, and the first 4 of those, give those kind of a casual shot, and I will send out, or I will do the same 4 worksheets, and then I'll scan those and make those available so you can see if you're on the right track. If you do those first 4 worksheets, there's way more space than a work effort there. I mean, there are Punnett squares and things, so it's not as monumentous as you might think 4 worksheets are. Then there are a second batch of 4 worksheets that I'd like you to do. That'll be your actual homework. And then I think there are 2 more sheets that are more difficult crosses, genetically speaking. And then that last sheet are the directions for doing your extra credit quadrohybrid cross. And by the way, if you're wondering what that's going to look like, this may not show you a very good picture of that, but it is quite the effort. We're using, we just made up some traits here. So there's a trait called A, B, C, and D for a quadrohybrid cross. And then I ask you actually to draw what the critter, a totally recessive critter looks like. But anyway, more on that later. So let me walk you through the basics of a monohybrid cross and a dihybrid cross. So when you're doing genetics problems, I've got a couple of targets here. So first thing you want to do is when a question asks you about crossing 2 parents, you've got to get the parent letters correct. So if they use the word hybrid for all traits, then it's big, small, or, you know, you're going to have a capital lowercase for each trait they discuss. If it's a monohybrid cross, it's just 1 letter. So let's say B, capital B, little b, crossed by capital B, little b. That'd be a monohybrid cross. A dihybrid, meaning 2 traits, you have 2 different kinds of letters. So maybe it's big A, big B, little b, and then the other side, big A, big B, little b. And keep in mind they can mix it up. So for instance, I might ask you a question about take a hybrid dad crossed by a totally recessive mom. Well, as you think about that, like, well, in mom, if she's all lowercase letters, could only make 1 kind of sex cell, so you can ignore the other 3 columns if you're imagining a Punnett square. So anyway, the second thing is to, once you've been given and correctly write out what the parents are for their letters, then your job is to how many different combinations of letters can they make? Like for instance, if we're just talking about 1 letter and you have capital B, you can only make 1 kind of sex cell. So on that Punnett square, just be capital B. If it's 1 of each, then you have 2 different choices for sex cells. So anyway, a dihybrid cross with 2 traits is kind of like the worst case scenario. So you're going to have to think of all the combinations of those 2 letters that could happen. And I'm going to do that for you in a minute. So we have the parent letters written down. We have figured out what sex cell combinations are possible. And the next thing is to actually build the Punnett square. And then lastly, you interpret, what those mean. How many are going to have the various traits? The dominant, recessive, dominant, and recessive. I'm imagining 4 different looks or phenotypes in a dihybrid cross. So let me cue you in a little secret. The answer is 9:3:3:1. That's the phenotypic ratio for any dihybrid cross. So if you're asked to do a dihybrid cross, you can just skip right to the answer. In a monohybrid cross, it's always 3 to 1. 3 that are dominant, 1 to recessive. So when I say it's 9, 3, 3, 1, it's 9 that'll have the 2 dominant traits, 3 that'll have dominant recessive, 3 that'll have recessive dominant, and then 1 will be totally recessive. So if you take a look at, here's my example. Let's pretend, per se, that we have capital N stands for normal nose, and lowercase n stands for giant honkin' nose. And let's pretend that capital T stands for normal teeth, and that lowercase t is a recessive gene for bucky teeth. And here are a couple individuals with those conditions. So normal person, okay, they got at least 1 capital N and 1 capital T. This person with the big honking nose, they got 2 little N's for nose, and they got 2 little T's for teeth. Bummer. Anyway, so let's cross 2 parents and see how this works. We'll cross 2 hybrid parents. So the first job is to, getting back here, is what are the parent letters? So it's going to be big, small, right, if they're hybrids. I want to do a dihybrid cross. So one parent is capital N little n and capital T little t, and the other parent is the same. Make sense so far? I wonder if you can even see that. Because it's written kind of high. So hopefully you can see that. Anyway, you know what? I'm gonna make sure you can see it. So those are my 2 parents. They're both heterozygous for each trait. Awesome! Anyway, next. All right, so once I know that, the second step is to figure out how many different combos can I have. And here's a trap that students fall into. They want to put big N, little n, and then big T, little t, and then they run out of choices. It's like, whoa, wait a minute. What you're saying there is a sperm cell or an egg cell has all the information for nose and then nothing about teeth, or vice versa. So every sex cell better have something about noses and something about teeth on both parents. So in other words, how many combinations of N and T can we come up with? And over the years, I kind of have a pattern set up. So there's some variability. Doesn't matter which parent you write across the top. It doesn't matter. And it doesn't matter what order you put these combinations, these possible combinations on the sides and up top. You just can't forget one. So it turns out the dihybrid cross is the worst case scenario. You'll have 4 and 4. So let's say this parent, well, let's see, I've got capital. That would be one sex cell, like a sperm cell. Another sperm cell choice might be capital small, then maybe small capital, and then maybe small. And if that came from that parent, then the other parent would be pretty much the same. And again, doesn't matter what order you list these. So big, small, big, and small. So all that's left now, I've got the sex cell possibilities. I fill out the Punnett square, and we'll see, maybe you're the genetic counselor and you've got parents in your office and they're saying, well, we know that we have this hidden trait for big nose and a hidden trait for bucky teeth and maybe cyclops, and so we want you to tell us what are the chances if we have a baby that they're going to be big nose and bucky teeth. We could probably have a kid that's got like the bucky teeth alone or the big nose alone, but we just don't want a kid with both. So, okay, well, my job as a genetic counselor is to tell you what are the chances. So I'm going to fill in this chart. So capital, small, capital. This is where having decent writing skills for neatness counts. I also am choosing to put the N's next to each other and the T's next to each other. There's no right or wrong there per se. So you could do the mixing if you wanted to. Capital. Let's see, capital small, capital small, capital small. I'm singing, and it's Saturday, and no one's in the building. Anyway, So what don't you want for letters? What would the letters have to be to have the big honking nose and the big bucky teeth? All right. We're done filling out the box. And so, now what? Well, the thing to do is to figure out what would it mean to look normal? Well, you have to have one of the two capital letters, right? So anybody that's got a capital N and a capital T, we're going to say that looks normal. So I think what I'll just do is put an X. I'll put an X in every sex cell combination that results in a normal looking individual. So that person's normal, 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 normal. Okay, so if you count out of 16, some number out of 16, so those that are the phenotypic ratio, those that are normal, normal, norm, and norm equals 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. That is to say they have at least one large letter, one capital letter for each the nose component and the bucky tooth component. All right, so how many would just have the big nose? Well, to have a big nose, you needed two small n's, but let's say how many have the big nose,